Hi, today I want to answer a very specific question, which is I'm asking for support for research on cancer in my lab. The obvious question is, why should we support your lab as opposed to giving money to, say, American Cancer Society or any other um deserving organization? First of all, my answer is you should give to anyone you want and everybody you want. But I want to tell you what distinguishes us from all these other worthy organizations. So I came to this country in 1977. I had just graduated from medical school in Karachi, Pakistan, and my idea was to cure cancer. Well, I started by studying acute myeloid leukemia and treating these patients. But within 7-8 years, it was very clear to me that given the complexity and the aggressive nature of this disease, in my lifetime, we will not be able to cure it. And I felt back in 1984 that the only way to seriously address the problem and try to help my patients would be to diagnose this leukemia early and to nip it in the bud. This idea came to me because I was seeing a group of diseases that are collected under the umbrella of something called Milosplastic Syndromes. So just as the name itself suggests, it's not any one disease, it's a collection of diseases. These are bone marrow diseases in which the blood counts fall because an abnormal stem cell population has taken over the bone marrow. And a third of the cases, one of the daughters will mutate and cause acute leukemia, and patients die from that. But in 2/3 of the cases where there is no acute leukemia, patients can still die within months. Why? Because it gives rise to complete bone marrow failure, and blood counts become so profoundly low that incompatible with life, and patients die of bleeding or infections. So I thought that maybe the biological issues in pre-leukemias or myelodysplastic syndromes would be less complicated than acute leukemia, and if we could learn to treat them, then at least we would prevent the appearance of this disease into acute leukemia. But then I needed cells to study if I was going to perform any serious scientific studies on them. And so, with that in mind, I just started saving samples on any bone marrows I did on my patients. So, blood, bone marrow, and for normal control, I would take their buccal smears from which we can extract DNA, and drawing their blood and separating T cells from them, because T cells are the memory cells, they live for like 100 years, and so we can use them as representative of what is normal DNA makeup for this person and compare them to the the DNA of malignant cells and find the differences. This tissue bank, which followed patients from MDS as they progressed to AML or progressed to the more aggressive form of MDS over years sometimes, has now mushroomed into the largest tissue bank in the country, collected by a single physician at least. We have thousands and thousands of samples from thousands of patients, and we have serial samples sequentially following the natural history of the disease. So after 35 years, how come I don't have any answers for my patients? And believe me, this is something that keeps me up at night. I feel ashamed to look at myself in the mirror, because it's true, I have not been able to help my patients as the way I should have. Everyone has excuses. Mine is that first we needed to assemble all these samples and now we have them. Second, we needed technology to develop. And third, I was also not sitting idle all this time. I've been working very hard to study limited numbers of samples for, say, the abnormalities in one or two genes or proteins. And I have collaborated with investigators from all over the country, whether it's NCI to Harvard to MD Anderson to Yale to our colleagues at Columbia, everywhere. Whoever wants to study this disease, I'm happy to work with them and do everything possible to help, because we are all working for the same goal, to find the right answer for our patients. And we have been publishing together in high profile journals, but with time it's very clear to me that what is really needed is not studying one disease or one protein, but rather taking thousands of patients and dozens of samples on each patient and studying them not just for genetic mutation or RNA expression, but for everything. So for example, genomics, proteomics, transcriptomics, metabolomics, panomics, every kind of omic we can come up with. We have to study these samples. So I have all the samples. Thankfully, the techniques have all been developed. But let me give you one example. To study 200 patients just by two techniques, which is genomics and proteomics, just to study 200 patients, we need $5 million. That's how much it cost. So I need the money to study 2,000 patients, not by two techniques, but by 5. Why should you give me money? Because I have spent 35 years collecting these samples, which are so precious, on a pre-leukemia that has evolved to leukemia. Our idea is we have the samples, we can trace back now from acute leukemia to MDS to what was the earliest sign of MDS, and then how can we start monitoring those people whose germline DNA that they were born with that made them susceptible to develop MDS. Who are these? Who has those germline DNA mutations? So the healthy population can start being screened for the appearance of the first cell in this way. So the question is, why should you give money to the first cell center and not to other deserving organizations? My answer is, I'm not trying to build a center where I will have 50 scientists and then I have to run around trying to raise $1 billion a year to support these 50 scientists. Absolutely not. What I'm asking for money for is simply to study the actual samples we have. And you're not giving money to me personally or to any company I have created. You will be giving money to the trustees of Columbia University. Everything is going to be done under the umbrella of the entire infrastructure of Columbia University. Every penny will be accounted for. And this is a very well-focused, well-designed, well-identified project with goals that are so obvious. It has to be done. So I think that the people who are at highest risk of getting cancer would be the best supporters of this center. Who are those people who are at high risk? People like my husband. My late husband, Harvey Chrysler, dedicated his life to studying cancer when he was 15 years old. And at 34, he got his first cancer. And then at 57, he got his second cancer and died after an almost 5-year-long struggle. He didn't die of the cancer. He died from the complications of the treatment we gave him with aggressive chemotherapies. So the people at high risk of getting cancer are those who already had one cancer. In other words, the cancer survivors. Today, in 2019, there are 16 million cancer survivors in America. Even if a million of them just give me $10 or $20 a month for 1 year, that is all that is needed to study this entire tissue repository for the fanciest, most cutting edge genomics and proteomics and transcriptomics and metabolomics studies, and find the footprints of the first cancer cell. I don't think that the investigators who are involved with me at Columbia and myself and all of us need to establish our credibility more than to show you our track record, which is that this is what we have devoted our lives to. This is what I have put thousands of patients through painful procedures of donating their bone marrows, and how beautifully they always tell me, Dr. Raza, even if it doesn't help me, take my extra bone marrow, take extra blood for me, it might help someone else. How humbling is that? We need to help our patients. This is why you should think about supporting the first cell center. Thank you.